So, when we made our introductions this morning, one of the things that we didn't point out, or at least perhaps didn't sufficiently emphasize, was ATAXIA's commitment to collaboration. And I am therefore delighted to be able to welcome Andrew Rosen from NAF to this conference, who has come all the way from the States, and Miriam Rye to co-chair with me this afternoon, who is from FARA, although not from the States. So we're truly grateful that they both felt able to be here, and I think it just serves to emphasize how important it is that we all collaborate together, and that this conference is a demonstration of that. So also from the States is Anne Newman, who is the Rare Disease Marketing Lead, Patient Advocacy and Engagement. That is a long job title. But anyway, that is her role. She has spent 20 years in nursing, so has a great familiarity with the needs of patients, and has spent the last 12 years working in patient advocacy with various pharmaceuticals, and currently is in a role with Biohaven, with whom we are delighted to be collaborating consistently, because she is coming to approach us and wanting to work with us. So we look forward to hearing your presentation. Thank you.
Well, I know we put industry at the end of the day here, so I'm standing between you and your fun evening. So I will try to be as upbeat and quick as possible with my presentation. Sue, thank you for that very nice introduction. Yes, I'm Anne Newman. I have been a nurse for a long time. I currently work in pharmaceuticals. I work for Biohaven, which is my distinct pleasure. And I'm going to tell you a little bit about us. Just hit the button. Arrow? Okay, there we go. So Biohaven today, and I'll tell you a little bit of why I called it Biohaven today. We are a high value with a lot of great platforms pursuing some novel paths for science to transform treatments of neurological and neuropsychiatric diseases. We have a very good portfolio, innovative portfolio. It includes our ATAXIA program, which I'm sure you want to hear all about. We also have neurodegenerative, neuromuscular, neuropsychiatric, and epilepsy programs ongoing as well. Now, we are a small company. We are based in New Haven, Connecticut, in association with Yale University. However, that does not mean we don't have a great deal of experience. My CEO, Vlad Korek, who is just a wonderful leader for all of us, we brought to market a migraine drug most recently, and it was a great success. However, he thought best for the community, the rare disease community, that we sell off that asset and give that to Pfizer and let them take care of it, and so that we could concentrate more on our neuromuscular and neurodegenerative disease portfolio. So that's sort of where the proven business model formula comes from. We do have a great deal of experience in bringing drugs to market. We are a patient-centric, we are a people-centric. It is my distinct honor, as I mentioned, to work for them and to be here with you today. Oh, these are in the wrong order. And these are the wrong ones. All right, we're going to work with it. I'm going to talk first about our ATAXIA program. So in 2017 or 2016, we started an ATAXIA program clinical trial of phase 3. It was not a global trial. In retrospect, that obviously would be different today, but back in 2016, that's exactly what we were recommended to do. Last year, it completed, so in October of 2022, we had top-line data come out that did not meet the primary endpoints. However, the team, our CEO, our clinical development team, our chief scientific officer would not give up on this program. This is a really important program to us. It's an important program for research for the community of ATAXIA. And so what we have decided to do over this past year is mine through the data. And so what that means is we have been looking through this data in all different forms. Originally, the clinical trial was for many different SCAs. It was for 1, 2, 3, 6, 7, and 10. And again, very difficult to have all of those meet a primary endpoint. But that being said, we have been mining through this data over the past year, and what we've identified is that we do see a signal, a positive signal in SCA3. Being the most common, that's probably not a big surprise to you. So our goal is for this year to engage with not only the FDA, but with the EU and the UK by the end of this year. So that's a very exciting time. I know that there's probably a lot of questions around that. However, I'm going to have to keep it very brief because I would not want to jeopardize any of the engagements that we have with any regulatory authorities. What I can say is, again, we are very dedicated to this community, and this is very important for us to move this forward because there isn't a lot going on in this space right now, and what we hope is after RIATA here, we're going to hear from another one of my industry colleagues, that will open the floodgates for more research. It's a step in the right direction. So I had a different, let me go back. It's a little out of order, but that's okay. We've talked a lot here about, and you've heard a lot about registry data, about the EuroSCA, about natural history studies, about disease burden studies. We recently completed a global disease burden study you may have heard of or seen on your websites. Many of you in this room may know someone who participated. Those results will come out sometime at the end of this month, next month, in different forums, so that's really great, and we thank the community for participating. But what I really wanted to talk a little bit about was why registry is important to industry and what that's helped us do. As I mentioned, we were not a global study, and when we thought about how are we going to get this particular program and the information out globally, how should we do that? Obviously, engaging with advocacy has been one of those things. But what we were also able to do was look at the registry data everywhere and where are the patients. Because we didn't have clinical trial sites in Europe anywhere, we didn't have any idea where are the SCA patients. And so I cannot stress enough how helpful the registry has been about what our plans are moving forward in engaging with regulatory authorities here by accessing some of that data, having the ATAXIA UK and other Euro ATAXIA help us sort of identify the needs and the unmet needs in the community. And that's really important. Again, I know we've talked a lot about clinical trials and everyone sort of is saying the same thing, but from an industry perspective, it's so important to us because it helps us identify where you are, what your needs are, and how we can help. And so thank you for participating in any of those trials. Continue to do so. Please, it helps when we think about what we're going to tell to regulatory authorities. It helps when we're engaging with different advocacy groups and things of that nature. Let me make sure I'm saying everything. And really, that's all I had to say today about our program. I'm happy to answer any questions that I can answer. This is our ATAXIA team. So Melissa Beiner is our lead investigator on this trial. She's been working on it with us since 2017. Again, a great dedicated team who is happy to be going through this process for such a long period of time and hoping to bring good news later this year. And I think that's it. I think I'm very quick today.
So our next speaker is already there. Our next speaker is Chantal Beekman. Chantal is senior scientist at Vico Therapeutics, and Chantal will provide an update on CAG, repeat expansion antisense oligonucleotide, or ASO, with therapeutic potential for individuals living with polyQ diseases. Okay. Thank you, Chantal.
Oh, just arrows. Okay. So, good afternoon. Oh, sorry. I'm short. I thought I'd look over it. No, it's fine. Trying again. So yes, indeed, my name is Chantal Beekman. I work as a senior scientist at Vico Therapeutics. We're a company developing therapies for neurological diseases. And VO659 is our developmental drug for treating polyglutamine diseases. So I'll just repeat some of the basics for you first. VO659 targets the expanded CAG repeat, that is the cause of these 9 polyglutamine diseases, and which includes the Huntington's disease and the 6 spinocerebellar ataxias, including SCA1 and SCA3. So the mechanism is that the CAG, the expanded CAG repeat in the DNA of these genes is used to make a protein with an extended stretch of the glutamine amino acid, and this makes a mutant protein that is longer and toxic and prone to aggregation, and this causes the progressive neuronal damage and then the well-known symptoms of ataxia. So with VO659 we hope to halt this process. So VO659 is an antisense oligonucleotide, in short an ASO. And this is an artificial sequence of RNA with some modifications to its chemistries compared to normal RNA, so it's more stable and more effective. And just to put it on the slide, here are the modifications of the that are, it's very sensitive, made to the sugaring of the nucleotide and also to the linkages between the nucleotides. So an ASO can affect their target in several ways, depending on the chemistry of the ASO and of the location of the ASO on the messenger RNA. So the messenger RNA is the copy of the DNA that is used to make the protein. So one way it can affect it is by affecting splicing, which is a part of the maturation of the RNA and removes the non-coding regions that are not needed. A second mechanism is the degradation of the mRNA. And thirdly, on this slide, it can block the translation, so the mutant protein is not made. So which mechanisms are the most relevant for VO659? So there are 2. If you look on the left-hand side, here in Huntington's disease and SCA1, when VO659 binds the repeat, so then by sticking to it, it's basically just in the way. So by steric hindrance of the ribosome that would otherwise make the protein, that's blocked. So the result is a reduction in the mutant polyQ protein. On the right, in SCA3, on the other hand, there's a different mechanism that when the ASO binds, the stretch that contains the CAG repeat is actually cut out by exon skipping, and this results in a shorter RNA molecule, and as a result of that, shorter protein that's lacking the polyglutamine repeat. So in total, this is a reduction of the full-length ataxin 3, but it's partly replaced by this delta polyQ isoform, so the ataxin 3 protein, which was shown by literature to still be functional. Moving on. So this was the mechanism, so now to the results, which is always the most interesting. Here, much of the early work on this ASO was done in Huntington models, and this is a Huntington mouse model where the mice were treated 6 times with an injection, ICV injection, so in the brain. And after 2 weeks, you can see we looked at the protein levels of Huntington, both the soluble and the insoluble aggregated form. And in these three brain regions, which are the most important in Huntington's disease, you can clearly see that there's a significant decrease of the protein depending on the dose of VO659 that was given. So we can find that in both different forms of the protein. But the most interesting part about this study was that we also looked at the motor phenotype of the mice, and we did it by using the motorator system, which is basically a tunnel that the mice walk through, but with cameras on three sides of the tunnel, which follow the joints of the mice and some other moving parts, so the movements can be precisely quantified. But because pictures always are better than words, I'll show you the videos. Here on the left, you see the healthy wild-type mouse that is just walking as mouse should, smoothly, relatively fast with the tail up. When you look at the model mouse for Huntington's disease, you see that it's much slower. It has these shaky movements and the tail is hanging down. But when you treat it with VO659, and this is of course the cool part, is that it's looking almost again like a healthy mouse. It's still a little bit more shaky, but it's pretty close to normal. So this made us very optimistic about the potential for treating Huntington's or, well, SCA patients also with VO659. So as I said, this is a Huntington's model, and of course we're here more for the ATAXIAs. So I'll start with a few in vivo results of ATAXIA models. So these are cells taken by fibroblasts from patients with SCA1 and SCA3, and we treated them in petri dishes with VO659. And when you see that, and we again look at the protein levels, that both in SCA1 and in SCA3 fibroblasts, we find a reduction of the mutant polyQ protein and also a bit of the wild-type protein. And importantly, you can see that the reduction is much stronger in the SCA3 fibroblasts. This is because of the different mechanism of action. And keep in mind that in this case, part of the full-length protein that is, removed is replaced by the functional truncated protein. So this is also shown in the next in vivo data. So this is again a mouse model. This time, this time it's a SCA3 model. And again, after 6 ICV injections, we see that in most of the brain regions, this time the one most relevant for SCA3, that there's a reduction again in the mutant ATAXin3 levels. And also importantly, if you look at the bottom part of the slide, you see the induction of the truncated isoform showing up in all brain regions. So this confirms the efficacy of VO659 in this second mouse model. We also looked in another mouse model, this one for SCA1, and this time, again, the ICV injections, only 8 of them instead of 6. And importantly, we also looked after 14 weeks, so that's like 3 months after the last dose, to see the durability of the effect. And in the graph here, we see not proteins levels yet, but the VO659 drug levels. And if you compare the green dots, so this is 2 weeks after the last dose, to the blue dots, which is, 3 months later, you can see, everywhere there's a clear levels of the drug, and that they're not going down that much. So this is, of course, good news, that the half-life is long, and you can see here that it was 136 days. So that's very positive, of course. What does it mean for the protein? So we looked at both time points, but I just show you the one after 14 weeks. Again, you see the green dots, a clear reduction compared to the mice that only got, we call it vehicle dosed, which means they only got artificial CSF as a control. And so, yeah, there is still in all brain regions a reduction again. And as a comparison, here you see wild-type ATAXIN-1, that's the actual mouse ATAXIN-1 with a very short repeat length, and that wasn't affected, well, basically at all. So it's very stable in mice, but we need to show that, of course, in another animal model before we can move to patients. That's still the regulations as far as I know. So here we have a non-human primate model. This is a cynomolgus monkey. These are healthy animals, so there's no polyQ mutant protein. And instead of the ICV injections, these are treated by intrathecal injection, so by lumbar injection. And after several doses, we looked 1 week after the last dose, and also in this time 6 months later. And again, we can clearly show the VO65 drug in all brain regions, although you can see that the levels are a bit lower for the very central brain regions, so the green is in here. But again here, the take-home message is that after 6 months, the drug is still clearly in there. And this time we calculate a half-life of more than 150 days. So this is very good. That means if you had to set up a dosing regimen, it wouldn't have to be very frequent, and you may be able to give it only every few months. But we have to see, of course. In this study, that was a safety study, we also looked at neurofilament light. So you've heard about it this morning as a marker that correlates with the disease severity or age or repeat length in SCA1, SCA3. But in this case, in these healthy monkeys, we're just looking at it if the treatment we do by the intrathecal injection of VO659 is not actually causing damage. And well, this is a good thing that we did, because you can see initially, after the first few doses, you do see an increase in NFL, which is an indication of damage. But as you can tell, it's not only in VO659, it's also in the vehicle-treated animals again. So that shows that it's due to the procedure of the intrathecal injection and not to the drug. And also, 1 week after the last dose, in both cases, levels have normalized again. And what's very important here is that after 6 months, the VO659, the NFL levels are very low again. And this, well, we just saw in the last slide that the drug levels in these tissues are still clearly there. So this shows that the drug is well tolerated. So with this good news, we're ready to go to the clinical trial. And in fact, the Arafico's first clinical trial for VO659 has recently started. So this is basically a safety trial with dose escalation, and we'll also look at some biomarkers to assess the efficacy. But yeah, indeed, it's mostly safety. It will include patients from 8 countries, up to 71 of them, and they will be recruited with 3 different diseases, so Huntington's disease, SCA3, and SCA1. So it's a basket trial. And the treatment will consist of 4 weekly doses of intrathecal injection. And at these times also blood and CSF will be collected, and also after a few weeks in the dosing period to do the biomarker analysis. So as this has just started, I cannot give you any results yet. If you want more information, you can go to clinicaltrials.gov and look it up under this number, or go to the VICO website, of course. With that, I will go to my summary. So VO659 is an antisense oligonucleotide that targets the expanded CAG repeat that causes polyQ disease. It has 2 mechanisms of action, either by inhibiting translation, so blocking protein translation, or by excising the toxic polyQ repeat from the protein by exon skip. The good biodistribution and long half-life in the monkey brain, combined with the lack of toxicity as judged by NFL levels in these monkeys, made us confident to go to the clinical trial. And in fact, we have started that, so that is good news. About the result of that, I can only say, let's hope it works as well in patients as it did in those mice. And I'll end with the one slide, the acknowledgements of the people in the VICO that performed the discovery work and made the oligos, and of course our CSO and our CMO. And also like to acknowledge the two groups, one at UCL and LUMC, that we are working together with on expanding our knowledge on SCA1 and SCA3 and seeing the effect of VO659. And of course, we'd like to continue productive collaborations with the consortia that VICO is a partner of. So that was my talk, and thank you for your attention.
We'll take questions now. Just to give you a chance to think about them, I've got a couple. Seeing as we just heard about VO659, I've never had a lumbar puncture, but I don't imagine it's very pleasant. And this is the way that you're looking to deliver the drug at the moment. I can understand that at the present you want to work out whether the drug works, but are you exploring any other means of delivery other than lumbar puncture?
Well, at this point, this is the way to go for us. Also, because of the localized distribution, and also it will decrease the chances of side effects if it's not administered by IV or something that's more systemic. So at this point, not that I'm aware of, we're not looking into other ways of administering it.
On the other hand, if it's as good as it seems to be, then people won't mind too much, I don't suppose.
I would agree. If it's that good as it is in the mice, then that would be good.
Yes. Triolose. The Triolose program looks like you think it's going to deliver because you're very optimistic about it. Are you, as a company, working on anything else in the ATAXIAs?
No, currently right now we are just working on this particular program.
I have one more question to Chantal. Thank you for the presentation. I was really impressed with the preclinical data, very promising. And I do have questions coming from the Friedreich ataxia field. I can't resist asking you if VICO has considered any preclinical phase or anything in the pipeline for FA with the antisense oligonucleotides, knowing that some approaches have been tried a couple of years at the preclinical level and were not conclusive.
Well, sorry, I have to disappoint you there that we don't have anything in the pipeline for FA at this moment. We are exploring the different ways we can use our ASO platform, and I can unfortunately not say in which directions this is going, but Friedreich's ataxia is not one of them.
Well, thank you. Any questions from the audience? Yes.
If everything goes fine and perfect, what are the time schedule? I know when I go back to our association and tell them that there are two companies doing a very good job, the first question will be, when do we get the injection?
So when it would be ready for market if everything goes perfect?
When can the patients?
This is a very difficult question because at first we're just really starting up now the clinical trials, which means getting ethical improvement and improvement of the local authorities in all these countries and starting up the sites. So that alone will take, I think, well, we're looking until next year probably when we can really expect real results, and then we have to do another study. So I really cannot give you, sorry, an indication, but it will be years still.
Is it 5 years, 10 years, 15, 20?
Well, I'm hoping more like 5.
That's what I want to hear.
Yes.
What about Biohaven?
So best case scenario, if we get positive sort of engagement from both the FDA and the EU. We would hope that a drug could come to market sometime the end of next year or the year after. But that's best, best case if they're positive.
Both of you, that's the hope I want to give my association. Thank you very much.
Any further questions? No, I'm sure we all want to ask you the things that you can't answer, so everybody's holding back. And in fact, extraordinarily, we are right on time. So I will call Vasilis to give his closing remarks. Thank you. So please, can we just thank the speakers one final time?
I think that we had a great day today. No closing remarks. Thank you all for your patience and your attendance. We need fresh air, of course, and so we move, let's say, our gala dinner half an hour later in order to give time to refresh a little bit. So let's say that we are going to start gathering by 7:30. 7:30. Okay. Thank you. Have a nice day.